All right, we're set to begin our next session. Ladies and gentlemen, thank you for returning. Thank you for coming at all. My name is Richard Hart. I'm not a scientist, but I play one on television. And you can see some of my reports on Channel 7 on Wednesday and Sunday nights on the 11 o'clock news. Mostly technology, but lots of science also. Tucker Hyatt, who puts on this wonderful event, is my long-lost brother. And we've determined that genetically because we both have 6 toes, and what are the chances? Okay. The next session is going to be, I had this great routine ready, but I have to skip it because we have to get back on schedule here. So this is about genetics. A little bit later tonight, by the way, these tables are here because we have the wonderful Wonder Cup Challenge. And the Wonder Cup Challenge pits, first of all, high school students against each other, high school teams from around the Bay Area for that little trophy over there, the Klein bottle. And then this evening, at the end of the next session, we'll all break for a little food and drink, and then we're going to seat professional scientists, professional researchers at one table, and high school students, professional high school students at the other table, and they'll be competing for fabulous prizes, and the prizes are big they're big every year.
First place, MacBook.
Okay. Second place, $500 per student. Whoa, is that right? Okay, and MacBooks for the winners. That's been true for a few years now. And one of the things that makes this possible, yeah, you thought second prize was 2 MacBooks. No. One of the things, I have to say, just a couple of things for promotional purposes and parking and other things. With the driver of an electric car, what are the chances? Who here has an electric car anyway? One of the things, how many people are here for the first time this year, have not come to a previous? Whoa, that's amazing. How many people are here because you got the postcard? Okay. How many people are here as a condition of your parole? Okay, a couple of people in the back there. Good, well, that's amazing. It's amazing. We're actually almost a standing room only crowd. We're doing something about the heat. I know it's part of the design of this room. We've got just opened up to the outside, a door here and one at the top of the stairs. So the stuff is rising out there, and I hope it'll get better in about 15 minutes. Apologize for that, but we're working on it. Okay, well, also in addition to this mind duel, we'll have the presentation of the Carl Sagan Prize for popularization of science outside. People have asked about these badges. You can get them from Eric Yao, a longtime supporter and worker with Wonderfest, who makes all sorts of variations on Picture-oo. You can go to the website, too. It's Picture with an R-O-O at the end of it. Okay, promotions, and we've got lots more. We've got comedians, we've got jugglers, we've got artists. But first, our next topic. We're at an amazing point in history right now with something called synthetic biology. And most of you are probably familiar with that all, but I'll just begin with one little story. I like to ask people a number of questions. I get to interview lots of CEOs of startup companies and some pretty smart people in this area, Nobel Prize winners and so on. And I always like to ask them a couple of questions. One is, where do you get your information? How do you stay on top of your field? And the other one is, if you were in the movie The Graduate, what would you say? Now, many of you are familiar with the opening scene in The Graduate. Benjamin just gets out of college, and his uncle takes him aside at his graduation party, and he says, Ben, I have one word for you, plastics. And he was probably right. At that point in history, he probably should have gotten into plastics. But I've asked a number of people in the last 6 months, what would you tell Benjamin today if you had one word? It's actually 2 words, synthetic biology. It seems to be a hot field not just for biologists, but for engineers who for some reason think this is an engineering problem and not a biology problem at all. But of course, engineers think every problem is an engineering problem. All right, let me introduce our first 2 presenters, and you will be able to ask them questions in a little while. They are both at the top of the game of genetics, but particularly in the kind of genetics we're going to talk about in this topic. Our question is, will genetics allow us to revive extinct species? Now, the first thing most of you think when you hear that is, Jurassic Park, we get a little bit of DNA that's somehow trapped in something through the years, and we can recreate a creature. And I have a suspicion that the answer to that question is yes and no. But as our last presenters told us, nothing is random. I'm hoping these 2 will be able to tell us yes or no. All right, one of them, our second speaker I will introduce first, is Ron Davis. Ron Davis is, to my mind, a historic figure. I've been following him since I first did my first science report on television about 20 years ago. And he is one of the people responsible for, I could go down a list of awards, but he has, among the most important to me were that he was elected to the National Academy of Science in genetics, and he has some lifetime achievement awards in genetics. He is the professor of biochemistry and of genetics here at Stanford and a member of the Stanford Comprehensive Cancer Center. And he has quite a web page on what he's working on currently, and I'm hoping he'll share some of that with us today, but it's some pretty amazing stuff. He's also responsible, and most famous probably, for Newton's fifth law, which is that for every parent there is an equal and opposite teenager. And I've always admired him for that. But one of the great discoveries in science he participated in and was responsible for in 1972, and that was the discovery of restriction endonucleases, which are essential for genetic splicing, recombining DNA. He's like the father of that to me. Now, he'll be more modest, but I'll try to get him to blush a little bit later. First, we're going to talk about our first presenter now, who is driven up today from Santa Cruz in a blinding rainstorm just to be here for you, so I hope you appreciate him. He is David Haussler. He is also responsible for a little bit of research, not as famous as Ron's, that listening to rap music over and over can lead to repetitive brain injury. But he's not talking about that today, okay? He is, as it says up here, at the Center for Biomolecular Science and Engineering, University of California at Santa Cruz, and is in a field, an expert in a field, which actually, when they did the human genome, there was a lot of math involved and a lot of computer science, and one of the guys who wrote the code for analyzing the code is this guy right here, Dr. Haussler. So he's also working on something bioinformatics is the field. I happen to know, this is quite coincidental, but among friends and family, a lot of people that I know have graduated recently or declared majors in colleges from Davis to Santa Cruz and Stanford. And out of a sample size of about 10, 5 are majoring or graduating in bioinformatics. Bioinformatics is a hot field along with synthetic biology. My personal bias, I think this topic right now is the biggest one of the 2 days here at Wonderfest 2008. So, without any further ado, from Santa Cruz, David Haussler. Ladies and gentlemen.
Thanks so much. It's great to be here. It's good to be here at a lighthearted event about some pretty serious science, and there's nothing more wondrous than the evolution of life. So I hope to be able to give you a tour of it, and if you don't like what I say, I understand they turned this machine on, and I'll stay away from that side of the room. Okay, so DNA. So you get DNA from mom and dad, and you get about 3 billion bases of DNA from mom, another 3 billion from dad, organized in the chromosomes. And there are 22 regular chromosomes, the autosomes, and the sex chromosomes, X and Y, that determine what sex you are. And they're all coiled up, and you see in this beautiful picture here, you can kind of see the way it's packed and can be unpacked. This is essentially a recipe for making a body. It's very profound. All life on Earth shares this common ancestry that life begets life, and that has been going on for 3.8 billion years. Each of these recipes is passed on from parent to child, and inside that DNA are genes, primarily genes that code for protein sequences that are the constituents of the cells that make up your body. It starts with one fertilized egg containing this message that you got from mom and pop. And somehow, those instructions are read out during embryonic development, and all of the organs of your body and the structure of your body and your brain spontaneously develops within the womb. And then you are born, and it continues to develop after that. So this amazing process is the product of this 3.8 billion years of evolution. And each of us carry this message in our genomes. How much does the genome determine? I always like to ask people to think about a pair of identical twins that you know. To the extent that they are similar, that's probably determined by these genetic instructions. To the extent that they are different people, they have different personalities, that's probably determined by their environment. So not everything that you are is determined by your genomes. Don't fall prey to genetic determinism. There's not a sense of hopelessness if you find something about your genome that you don't like, and you may very well in the next few years find a lot of things out about your genome. We don't believe in genetic determinism, but nevertheless, it is profound how much information is contained in your genome and does determine you in that sense of structure, form, the basics. We were very, very proud at Santa Cruz to be able to put up the first working draft of the human genome. This happened on July 7th, 2000. And it's all due to the efforts of scientists like Ron Davis and others in 20 labs across the nation who put together sequences of DNA that they had sequenced with the new, at the time, sequencing technologies. And we were able to write some computer code, mainly Jim Kent, actually a student at Santa Cruz who was really the genome hero, wrote the computer code that put together this first draft of the genome. And I always like to show this slide. When we posted that public working draft of the genome at July 7th, we got an amazing amount of traffic. This plot is total outgoing internet traffic from the entire campus of University of California, Santa Cruz. And you can see the peak that we got at this point dwarfs the total traffic that had occurred and was typical for the years preceding that. 500 billion bytes of information were broadcast in a 24-hour period out of the Santa Cruz campus that day when everybody wanted to read this recipe that had been passed down for 3.8 billion years and determines the genome of one person, one reference genome at that time. Now, can you get away with just one reference genome? Well, people sitting in this room, your DNA is 99.9% identical to a typical other person sitting in the room. If it's your son or daughter, probably much more so than that. But if we have that one sequence, we know enough then about the typical genome sequence of a human individual to start to wonder, essentially, at all of the complexity and the genes that are contained within it. Rapidly, we were able to sequence genomes from other species and compare them. And now, if you go to Santa Cruz, you can see this, and I encourage you. There are lots of amateur genome sleuths that come to our website. It's genome.ucsc.edu, and you can poke around at the reference human genome, the reference mouse genome, the reference rat genome. Now we have almost 44 different species of vertebrate animals that we have draft genomes for, and you can compare them. You can look at the gene and see how it's different in the other species. And you can look at a lot of information that we gain about these genes by doing various molecular biology tests and doing genetics, essentially. There are about 7,000 users that we get every day. Now, these chromosomes that you see when you sequence a reference human genome are the product of a long process of evolution. So at the top, if you see, these could be 2 ancestral chromosomes containing hundreds of millions of bases of DNA. And the things that you see at the large scale are changes in these chromosomes that happen. And most of the changes that we see that are the result of the evolution over millions and millions of years started in one individual. That's very hard to comprehend, but it's a random change to the DNA that happens in one individual and then gets passed on to that individual's children and their children's children, and eventually everybody in the population has this new version of the genome. Most changes don't have that fate. Most changes get started and then die out. They don't really get passed on to many people. But some changes do, and that's how genomes change over time. So these changes, let me go back here. These changes that you see can be of the type where a big segment gets duplicated. You see the purple segment gets duplicated. Now we have a whole bunch of extra DNA. You can swap the ends of 2 different chromosomes. Now, after the species split off, they're going to have independent changes. So the species on the left has a chromosome fission. Now it has an extra chromosome. Species on the right had an inversion. A whole piece of chromosome is inverted. These can create new combinations of DNA then that do different things through the ages. Now, if we look at the genomes of the extant species that we have sequenced so far, we see amazing similarities between them. We see hundreds of millions of bases of DNA in chromosomes, and we see stretches of millions of bases of DNA that are very, very similar between species. Even between different mammals like rats and humans and dogs, we can see stretches of chromosome that clearly evolved from an ancestral piece of chromosome and had these independent changes after the species diverged from each other. And by taking those and reconstructing a common ancestor for them in terms of the chromosomal shape, we can get pictures like this. And this is work in my lab recently where we've reconstructed the evolutionary history of the rat X chromosome. Each of the ovals represents pieces of DNA that have not been chopped apart by these big rearrangement operations that I was showing. And you can trace them from their ancestral form through these lines of descent, and you can see how big pieces of the chromosome have been moved from one side to another. Some of them have been inverted, like this purple one. And you can even zero in on pieces of this and zoom in further and see more and more detail. Here's a little piece which you didn't see before, which is inverted. It's hard to imagine the scale of this. We start with hundreds of millions of bases, and as you zoom in, you can go down to changes at very small scale. Now, what happens is these large-scale changes in the genome do produce new capabilities sometimes by essentially creating new DNA structures, new genes. The typical way that new genes are created is by what's called segmental duplication. And this is just a really quick schematic of this. Evan Eichler, in general, the credits will be down in the lower right corner, so do look at those. The ancestral configuration for the great apes genomes is exhibited in this little diagram of the baboon chromosome 3. And you see that there are 4 different segments of DNA, which we've just colored so that you can keep track of them. These 4 segments of DNA are ancestral pieces of DNA that were doing something in the baboon genome. In particular, this lime green one has a gene called Morpheus, which is an important gene. It exists in the nuclear pore complex. We don't know exactly what it did. But in fact, we see that in the humans, because of segmental duplications, these pieces of DNA have been copied and rearranged to create this amazing array. And so we have all of these different copies of Morpheus, one in each of these lime green blocks, and they've landed on human chromosome 16. So we actually have genes that our ancestor didn't. And this is often a way you get new functions. One of these genes might be doing what the ancestral function of that gene was, and the others essentially are free by evolution to evolve into new functions, new capabilities. The opposite of that is genes do get lost as well. And here's an example now where we've jumped down to the individual DNA level. So this is an actual piece of the A's, C's, T's, and G's on a human chromosome that we've traced out. And by comparing that piece to the genomes of these other species that you see here, we have predicted how it evolved. Now, as I said before, what we find are these stretches of DNA, much longer than this one, that are almost identical between species with just a few changes. And by tracking those changes and essentially running them backwards, we can deduce what this stretch of genome looked like 6 million years ago at the time that the chimpanzee lineage diverged from our common ancestor, 14 million years ago when the orangutan diverged, and so forth as we go back in time throughout these mammals. Now, the green part is part of a protein coding gene. It's very important that proteins are coded by reading the bases 3 by 3. So you read 3 bases of DNA. That has to be transcribed into RNA, and then it will make 1 amino acid when it's translated into a protein sequence. And so as you read this 3 by 3, that is the code for the protein sequence. But there's one special, well, there are 3, but one of the special codons is TGA, and that doesn't code for an amino acid. That codes for stop. And that indicates to the ribosome, which translates the RNA into protein, that you should terminate. So this change, which we can isolate in this whole diagram, where this G changed to an A just about, say, 7 million years ago, before chimps and humans went their separate ways, killed this gene, essentially, from an evolutionary point of view. This gene is no longer functional in chimps or humans, whereas it is in all of these other species and in species as distant as bacteria. It's a very, very basic gene, acyltransferase 3. It's an It's an enzyme that performs a fundamental biochemical function. And when you see this example, think about what is the fate going forward. We can't predict evolution going forward. Lots of people always ask me when I give this talk, well, if you can predict what happened in the past, can you predict what's going to happen in the future? And the answer is no. And I think Daniel Fisher explained a lot about why that might be hard, right? These initial conditions could form very, very many different fates going forward. But one thing I can bet on is that we'll never reconstruct this gene unless we do it by engineering. In other words, it's very unlikely by chance that we would have the reverse evolution and somehow recreate this enzymatic function. Before you know it, when a gene dies like this, other mutations happen because there's no Darwinian selection to keep the gene going, and then the gene just fades away. And it took 1 billion years to invent this enzyme, and sadly, we lost it, and we won't recover it by any kind of natural evolution. Here's the last example, and this is a gene we're studying in my lab. Speaking of Darwinian evolution, you can see the traces of Darwinian evolution when you line up the DNA. And here's a segment of DNA on human chromosome 20, where we've lined up the corresponding DNA from all of these species, even from this species as distant as chicken. We have this remarkably similar DNA. And this indicates that there are very, very few changes in this region. This is part of a 120 base region, which only has 2 differences between chimp and chicken. Why would that happen? Mutations are happening all of the time. And if you look at the typical piece of DNA, it's radically different between these species. But in this case, changes were probably detrimental to the fitness of the organism. And so they were eliminated by negative selection. So we're getting changes all of the time, and Darwin's theory predicts that changes that reduce fitness won't be passed on to future generations. Changes that are neutral or increase fitness might be passed on to future generations, and to the extent that they increase fitness, are very likely to be passed on to future generations. And if you make a random change in the genome, it's bound to decrease fitness. It's hard to take this beautiful machinery of the genome and randomly change it to get something that actually will improve the fitness of the organism that carries that genome. And so when we look at the genome and we think about Darwin's theories, we see them manifest in these remarkable regions that are unchanged. All of your protein coding genes show this, and at least as much material as contained in the protein coding genes times 2 also contains this. So there are other things in the genome that show this remarkable negative selection besides the protein coding genes, and this turns out to be one of them. This is a gene that makes an RNA, and it's very exciting because after being conserved for hundreds of millions of years, since the common ancestor with chicken, it changed in 18 different places in the human genome. This is the most rapidly accelerated region in the human genome, and it led to the discovery in our lab of a new gene that turns out to be involved in brain development. So we're currently examining that gene. We don't know exactly what it does, but that's exciting, and that's something, a remark for your amateur genome sleuths. These data were on the genome for a good 1.5 years on our browser. Everybody was looking at them. You could have found it before us. I'm serious. We were lucky to find this. Okay. We currently have a project to reconstruct the ancestral genome configuration for the placental mammals. Here you see the sampling of placental mammals. The red ones are the ones that you can see and download the genomes for on our browser. The blue ones are ones we'll get soon. And if you go back, you can see at each one of these branch points is a genome that was the common ancestor to everybody that lies below it. By looking at these genomes and comparing them and looking at the differences and similarities between them and the outgroups, the neighboring genomes, we can reconstruct computationally the genes in these ancient genomes with approximately 98% accuracy. And so if we go back, the first thing we start with is the human genome. And then if we are successful, a very exciting event will happen. Svante Pabo's group is getting DNA out of the ancestral remains of the Neanderthal, a 38,000-year-old tooth is yielding good DNA, and they hope to sequence that DNA and give us a glimpse at the Neanderthal genome. Now, 700 to 800,000 years ago, there was a common ancestor of human and Neanderthal, and by comparing the human and the Neanderthal genomes and using the outgroups, we should be able to reconstruct what that genome looked like. But then we'll just go further, and we'll reconstruct what the genome of the common ancestor of the chimps and the hominoids looked like. Gorilla, orangutan, and as you go back, you can get more and more ancestral genomes at each of these points in our evolution, going back further, about 40 million years, and so forth. And you can see, at this point, we are looking at the common ancestor of a fairly diverse set of species, something that was the common ancestor of the tarsier and the other monkeys and apes is quite something distinct from what we would see today. And yet you can always see a bit of a common pattern. And I've chosen animals that are more and more representative of what we feel from paleontological evidence was the ancestral form of these common ancestral form of these species. In particular, our ancestors were shrew-like. And you see that there are more and more species that look like, as we go back here, the common ancestor of all of these species was probably similar to what we see in the common shrew today. And if we even include now all of the placental mammals going back 100 million years, we would also include the common ancestor with this guy. We want to reconstruct, and we can computationally reconstruct, the 14 steps that lead from human back to this common ancestor. A snapshot of the genome as it existed at each point along that backwards trail. And then, to the extent that we have more genomes here, we can take that ancestor and run it forward and look at the changes that happened to it as it then created the other species. Now, we can't go back forever. At this point, it gets harder because the next convergence is at 180 million years ago, where the marsupials and the mammals diverged. It'd be difficult to reconstruct that far from a computational point of view because too many changes have happened with no intermediate species. But it would be great, and we can do that. It would be great for some genes to be able to do that. We can do this for a subset of the genes, and for a subset of the genes, we can even go back 300 million years. But returning to the placental ancestor, already we have a very exciting thing. How did you get from a shrew-like common ancestor 100 million years ago to elephants, dolphins, bats? All of these are evolutionary changes, and they are traceable to changes in the genome. So in the next years, we will actually be able to document those changes. We will be able to see how the A's, C's, T's, and G's changed from an ancestral form of the genome to a modern form of the genome now that is capable of being a recipe for these vastly different mammals with their different forms. That will be very, very exciting. And of course, it brings up the topic of this meeting, and I will close on the next couple of slides. Could we actually resurrect these ancient species? I hope I've convinced you that we can computationally approximate what their genome looked like. And I've told you that a genome is a recipe for the species, for an individual in the species. So how do you actually create that? And I'm going to defer to my expert colleague, Ron Davis, to tell us about the biotechnology that would be involved in actually taking that genome sequence and resurrecting an individual representing that ancient species. But I will say that there is a possible slow way. And Gisela Ciccone from Yale recently found a very interesting relationship when she was looking at the giant tortoises on different Galapagos islands. It turns out that there is an extinct variant of the giant tortoise that Darwin actually observed being slaughtered hunted essentially into extinction in the 1800s, that lived on Floriana Island. We can still get DNA from that ancient species. So even without reconstructing what we think that species might have looked like, computationally, we can, in this case, go directly to the ancient DNA, sequence it like we're doing with the Neanderthal, and find out what the genome of that species looked like. Now, what Gisela suggested was not that we actually biotechnologically reconstruct this, but that we look for other related species. In particular, we look at the giant tortoises on Isabella Island, a related species, and see if some of them carry these gene variants that we find in the extinct species. And that was a very good idea, because these variants persist for millions of years, actually. And so for hundreds of years, you're bound to find variants that are similar for millions of years, you are bound to find variants that would be common between the two species if they diverged on the order of 1 million years ago. And so those species would still be segregating, those genes could still be segregating in individuals on Isabella Island, and they are. And so in this sense, the genetic information from this extinct species is still alive as rare genes or uncommon variants in this other species. And so she suggested that we actually breed these individuals in a very concerted manner to force them to gradually become what the extinct species was. And it's a fascinating idea. So there's a path where we might actually revive an extinct species. We know the genome. We know where we want to go. We have a related genome, and it has lots of the material that we need, so we can maybe push it. I think that is a slow way of doing this. But it's an interesting possibility. So we'll make up a name for it, the Phoenix Project. So could you do this crazy project to bring back some of these ancient forms or the extinct sister species of these ancient forms? Maybe you could do it by gradual breeding, but maybe with advanced biotechnology and synthetic biology, we could greatly accelerate this project. I think it's a fascinating question. Let me just, before I turn it over to Ron, thank some people in my lab. Sophie Salama is my wet lab director, and she's responsible for a lot of the work investigating the human accelerated region 1. Jim Kent, I told you about, a hero of putting together the first draft of the human genome computationally. Adam was a pioneer in these highly conserved regions, as was Gil Bejarano, who's now a professor here at Stanford. Adam is a professor at Cornell, and Katie is a professor at UCSF, and she computationally beat you guys to the punch. She found that HAR1 gene before anybody else did. All right, thank you.
Just take a moment here to say a word, some extra about Ron Davis here. He's working on a couple of things which are not really very focused on the topic today, but I'd like him to talk about. And I would like somebody here to ask him, what is an oligosynthesizer? At some point. It's the
I'll show you a picture of one.
Okay, all right. And the other thing I have to say about him while he's speaking here, he probably won't talk too much about this today, but we almost had him talk about his interest in health care for you and for other humans on Earth. That he's concerned that there are ways that we can use the genome to better treat people and the information we know about that. So I'd like to direct some discussion after this about that. It's all genetically related, but he is a many-talented individual, and I'm hoping we can get into most of what he knows today. Okay, ladies and gentlemen, Ron Davis.
Thank you. So we're doing this as a team, and his was the introduction and all the information, which is also his specialty. And I'm a chemist, and so and a biochemist. So I work on trying to solve the biochemical kind of problems. So I put together some real simple slides just to keep things focused, because I didn't want to get into a lot of detail and a lot of chemical structure. And what are the problems? And first of all, I'd like to start out, what do we know already? What happens when you get into a new field like synthetic biology is people are gung-ho and they go in and they start solving problems, and they don't realize that that problem was solved 40 years ago because they don't read the old literature, and it's hard to find. Eventually, we'll have it easy. So the basic the basic problem here is, what do we tackle and what do we focus on? This is central dogma of that the DNA that we've just been hearing about, and it makes RNA, and the RNA makes proteins, and the proteins are the building blocks that make up cells and so forth. So this makes things real easy because we can just simply focus on this part, and we don't have to deal with the other parts very much, and that helps this whole problem a lot. And the other nice thing is DNA is very stable, and that's why you can get it out of these ancient organisms. That's a remarkable thing about DNA is its chemical stability. RNA is not. And it's possible that RNA was the beginning of the genetic information billions of years ago, but it has a problem. It can easily degrade it. And so one of the, probably in early evolution was the development of this molecule, which is chemically much more stable. So that makes it easy. Now, so where are we? And that is, now this goes back 40 or so years ago, 60 years ago, is that to know that we can take bare DNA, purified chemically, and we can do a transformation, that's the word that's been used, to put the DNA into the cell, and we find out that it works. And so that says a lot of the information is just encoded in the DNA. We don't need extra stuff. That simplifies the problem tremendously. Okay, so another development, this is some 30 or so years ago, is the development of a technology called recombinant DNA. And why that's relevant is that technology allowed us to splice together pieces of DNA, and it's kind of the forerunner of synthetic biology because we're taking DNA and we're doing manipulations of it. People got very concerned about this technology, very excited what it could accomplish, and also very excited about what evil things could be done with it. So there's an awful lot of hysteria was created at the time of this development. So far, nothing bad has happened, but the clock's still running. And this technology said that we could take foreign DNA and something called a vector that's allowed it to be maintained, and we could put it into another organism, and we find that this foreign DNA works. So that means that I can take DNA from one organism and put it into another, and it still will function okay. Now, there's a lot about functioning that sometimes is details that you have to worry about. But in principle, it will function. And in fact, this is sort of the basis for a lot of the biotechnology industry. You take a human gene, and you put it into bacteria. You have to do a little manipulation to make this all work. And you can make the same protein that is made in humans. And in fact, that's the basis for a lot of therapy. You're missing a particular protein. You can make it in bacteria. You can put it into people. And it will work there. So, and this is generally true. Whether I put this foreign, I can put this in a number of things. I can put this into bacteria. I can put it into a fungus. I can put it into a plant. I can put it into a human cell. And it will basically work. And the reason for that really is that all organisms are basically the same, with only differences in sequence. And so the rules of how it works are all the same. And that helps the problem tremendously. So these are the old problems solved that we don't have to solve again. Another thing is that I can chemically synthesize DNA. And the chemistry is a little complicated. I was toying about showing you the chemical steps, but no, I don't think it's necessary. It's complex chemistry. It's difficult to do it because you have to get rid of all traces of water, which is obviously hard, especially on a day like today. And you can't get any oxygen present. So you have any trace of oxygen or water, it doesn't work. And so that makes the synthesis difficult and the instrumentation difficult. But the chemistry was well worked out, and you can chemically synthesize a piece of DNA. Now that's very valuable because you can take the databases that you've just heard about, and I can take any of those things and I can synthesize them. I don't have to go get them. And in fact, if he says, well, this is a sequence that must have occurred, but we don't have any examples of it because we can't find any of that DNA, but it must have been there, I can make it. I can make it directly from the database. And we know that if I make a human gene here, because I know the sequence, I can put it into a human cell or bacteria cell or a yeast cell, and it will work. So there's nothing magical about the fact that it's organic. in the sense that it has to come from a natural source to make it work. I can strictly make it from chemical means, and it will also work, and there does not appear to be any difference. So that's a good one because it says there's no modifications of the DNA that it hadn't yet discovered. And that would be possible. And so we can make synthetics. Now, this is an example right here, which we're already now into synthetic biology. And so when you hear about synthetic biology, a lot of it is this right here. Since I can synthesize things, I can try out things. So let's suppose I wanted to try to make a whole bunch of different proteins. Or I wanted to make a protein that I found it was present in a genetic defect of an individual. I can take any of those things and make them. And I can make proteins that never existed before because I can design it and make it. So there's a lot of synthetic biology at this level. One gene, put it into some organism. A lot can be done there. And sometimes you can expand this a little bit to gene shuffling, and that is, well, I don't want to do just one gene, I want to do 10 genes. And I want to see how they work together. Well, that's also possible with this technology. That's not so hard. And so there's probably the bulk of activity in synthetic biology is this. But this is not really what we're talking about here. We're talking about something much more complicated. We're talking about sort of how to resurrect an extinct organism and not just deal with one gene. So their problems go on. Now, another thing that we learned about 20 years ago, these are all things that you don't have to rediscover. And that is you can make artificial chromosomes. Now, you saw that with human. Not a human artificial chromosome. That's much more difficult. But I can do one for yeast. Now, yeast is an organism that's very similar to human in many respects. Many of the genes will cross-function. You can move them back and forth. They'll work just fine. But it's a much smaller genome and much smaller pieces of DNA, much smaller organism, much easier to work with. And it was discovered then that, in fact, you could put a lot of sequences there from, say, human, and all I had to worry about is getting pieces of DNA here that would cause replication, so I can replicate the DNA, and there are specific sequences that do that. I had to put things on the ends here to be chromosomal ends on both ends. And then I had to put another sequence called a centromere, which all organisms have in these linear chromosomes, so that it would segregate properly. And we know that those sequences are too. And so those can be put in, and now I have an artificial chromosome. Now, obviously, it has a lot of different proteins that have to bind to that synthetic chromosome. It has to have histones bound to it. It has to have the centromere reconstructed. The ends, the telomeres have to be reconstructed. And these orders of DNA replication have to be there that they will work. So there's a lot of things that bind to it. So a lot of people sort of say, well, that's impossible. How would you get that all to work? It's easy because it's all encoded in the DNA. I don't have to do anything. So people think this is a real problem. It's not a problem at all. All you have to do is take this DNA and get it into a cell, which is hard. That isn't the problem. But once it gets in, everything's reconstructed. It's all encoded in the DNA. So that's not a problem either. And as we go forward, that probably will not be the thing that hangs us up. So how do people go about doing more complicated synthesis? And this is where we start to get to the edge of our abilities to do things. And so when we synthesize DNA, the chemistry is great, but not phenomenal. In the sense that I do it just like I would do normal DNA. It's these bases, and I have these individual units, and I can do unit 1 and then 2 and 3, and I can build it up. Okay? The problem is that it sometimes doesn't work. And so there's a limit to how long I can make it. And so in the past, the limits were around 40, maybe to 80. Were the limit. That's as best I could do. In fact, I'd have to purify that DNA and try to get it away from all the stuff that was all the junk. And now we're doing better. We can actually make things around 200 bases long. So those are those individual A, G, C, and T's that go in the DNA. Now, DNA is double-stranded. There's 2 strands, and they're complementary. So I have to worry about that too. And so what's usually done, and just as the usual, is you build molecules, individual molecules. These are in this range of size. Not clear yet what works the best. And then I make the complementary strands and overlap them so that I can build this up. And either I leave no gap here or I leave a gap. Both of those are a possibility. And then I use two different enzymes. And matter of fact, these enzymes were famous because professors of biochemistry here at Stanford were the ones who characterized these two enzymes. And one of them will fill up the hole, and the other one will join the two pieces together. And then I have a long piece. So that is still synthetic, but I use some enzymatic help. And I can build up a gene then. And you can do this now. This kind of thing can be done commercially. And so there's a number of companies. So the length of those pieces is around of a gene. So I'm going to build a gene. I don't have to go into the laboratory and make it. I can order it. And I tell them, here's the sequence I want, and I can order it. Now that's about 1,000 bases to 10,000 bases. That's what you can order, roughly. In fact, one of my old students has set up one of the companies that does that. And this is what they do with variations on this thing. And the other thing that they'll do is that once they get it all built, they'll resequence it and test it, make sure it's right. And if it's not right, they'll do it again. So what you get back is what you asked for. Now the cost of that is about $1 a base. So if I want a 10,000 base gene, it's going to cost me $10,000. Now that stops being something that an amateur might easily do. Because that's all you get is the gene. You get nothing else with it. But that's where we are at the commercial sector. And people will use these commercially available components to do larger projects if they have enough money. So there was a recent assembly of a bunch of sequences put together, and those are all just ordered. And if you have $1 million to spend, you can put together 1 million bases. And you just order up the pieces and you stick them together. So we're at that stage. If you have a lot of money, you can do quite a bit. But it gets still pricey if you want to do human. You know, that's $3 billion. I haven't ordered it. So now let me go into more the real problems that we're facing. Now let's suppose we take on something. We want to do an extinct species. Bigger. Probably out of the realm of this commercial. We've got to get into the real problems. So, and I'm just going to give you a list of the problems. And this will, and some of these are somewhat solved, some are not solved. And this is what someone who's trying to develop technology or trying to do these kinds of problems has to face and not ignore. And that's where it makes it not trivial. One of them is that the synthetic DNA monomers, these are the pieces that you make, for a very long time, they were obtained from natural sources, probably beer yeast, because you have a lot of yeast left over and it's full of these, and you simply then purify them. And then you chemically sell them. The problem with that is the purification isn't great. And when you get it, the one monomer out, it's cross-contaminated with all the others. Well, no one ever worried about that in the past because they weren't worrying about gene synthesis. But if you actually use those for gene synthesis, you get about 1% errors. The wrong sequence goes in because it was in the raw material. So that's a serious problem. 1% is a lot. Now, since then, what's happened is that, and realizing this serious problem, what's happened more recently is that they had to chemically synthesize the monomers. So there is no you can't purify them better than that. So there is no cross-contamination. And those are now available. So that gets rid of this problem. And this is fairly recent. Another one, this has not been solved, is that when you add these monomers one after another, on one molecule, occasionally, the reaction fails to happen. And now you go on to step 2. But you didn't finish step 1. and so it's missing, and it's a deletion. So a base is removed. If you know anything about how you read the code for like coding regions, you read them as 3 bases, and then you go over here and read 3 bases. What happens when you miss a base? You're out of phase, and it's gobbledygook. And it will not be functional, or it will not be the function you're looking for. So that's a real, and that's worse than making the wrong base in. The wrong base, most of them probably won't do anything. A missing base is a disaster. And so you have to work very hard not to have this happen, but it still happens. So that problem is not solved. So therefore, we're probably not going to solve that problem. And you're probably going to have to do something called error correction. Nobody has really perfected error correction. Now, they've done things to improve things. They've gotten some strategies which get rid of some of the errors. And then they rely on sequencing and then throwing out the wrong ones. But when you get very big, you can't do that. So a very robust error correction strategy, proofreading strategy, has to be developed. It would be equivalent to, I'm going to proofread my paper line by line and find mistakes. You will not get them all. Or I have a computer do a scan. And at least if you code it right, it will find everything. At least it will find everything it's supposed to find. And that level is what we have to go for. We have to go to something. But it's not going to be at the computer. It's going to have to be biochemistry. We have to do some biochemical step that we can identify mistakes and eliminate them, and probably do it without a lot of effort. If you have a lot of effort, it's going to cost too much. So it's going to have to be automatic. So the error correction problem still has to be solved. Another one is misassembly. That is, when I showed you all those pieces you have to put together, sometimes you make a mistake and the wrong piece goes in. Now that's more than a single base mistake. And your error correction mechanisms may not catch those. So you're going to have to do something to make sure that you find any misassemblies. And the misassemblies sometimes are actually a real serious problem, because it almost always gets misassembled. And we don't know the rules yet. If you just simply put in those pieces, little pieces, and put a whole bunch of them in to try to make something very big, you almost never get what you're looking for. You get something else. And because it doesn't work the way you think it should work. We don't know the rules for that. Why does it misassemble? And so the bigger you make it, the more pieces you try to assemble, the more mistakes are made. And people have worked on this problem because there's several companies that are doing this commercially. That is their biggest problem. If I could just make all these pieces, put them into a tube, assemble them together, and I'm done and I can sell it, boy, that would be a piece of cake. No, they can't do that. They find it misassembles. They have to go back and figure out where it misassembled and then try to make a correction. A lot of labor. So that is a problem that has to be sorted out a lot more. Another problem is really size. Now, when I've showed you the synthetic biology where people are, these 1 gene pieces or something of that type, even 100,000 bases put together, that's not a size that's a problem. However, when you get to 1 million, and we're talking many millions if we're doing mammals, when it gets to 1 million, then just the flow of any movement of the liquid, in other words, if I put it in a test tube and I did that, I would break it to pieces. Because the molecules are very long and they're not that strong. They're incredibly long polymers, longer than almost any other, than the plastics. They're much longer than the plastics. And that makes them easily broken. So this is another problem. We have ideas about how to solve, hasn't been totally solved. We're going to have to deal with that. Now, there are several ways we could try to deal with it. One would be that, well, okay, I can make 100,000 in the test tube, and I'll make sure it's right by these various biochemistry, and then I got to leave biochemistry because I can't handle the size. I've got to put them into a cell. Now, what kind of cell am I going to put them in? Well, I don't know. If I'm going to try to do something equivalent to a mammal, then I'd put it into a mammalian cell. But boy, they're hard to handle. And then once I get it into the mammalian cell, how do I get the next piece hooked to it? Not so easy. We don't have a good way to do those things. I could put it into a yeast, and I could rely on yeast. We'll put the right pairs together, and I could assemble it in yeast. And that's probably what's going to happen. At least that's our plan. And I can go to 1 million in yeast, maybe 4 million in yeast. But then that gets to be almost, the whole genome is only 12. So I can't put much more than that in there because I'm going to wind up with the wrong DNA sequence being the bulk of what's in the cell, and it will probably eventually die. So I can go from 100,000 to maybe 4 million. But then I got to build something bigger than that, and we don't have a clue. We'll have to figure it out. We'll have to figure out some other organism that we can then move it to and do the remaining building, or we'll have to take mammalian cells and engineer them so that they can do the assembly correctly. The reason I'm saying it's not trivial is that the mammalian cells are famous for misassembling. And they just will take random pieces of DNA and bash them together. And they don't take things that they should recombine and do it very well at all. And so trying to do this in a mammalian cell, I'll lose ground. I'll be making more mistakes than I'm correcting. And so we have a serious problem there too. I think it's solvable. And we have to kind of bootstrap our way into it before we can actually face it. But I think it can be solved. It's also possible we could figure out some clever biochemistry way. The cell obviously can handle big, big pieces of DNA, and it doesn't shear them. And so we might, in fact, say, okay, I have to kind of build, I don't want to use an existing cell to do this in. I want to make an artificial cell to do this in. And it takes all the features that are present in a real cell to keep it from breaking. But it doesn't have all the other, it isn't alive. So the problem, if I try to do this in a living cell, I have to keep the cell alive. Now that can be good because I can grow it and make more. But it also has to be viable, and it has to carry out all these functions. So if I make an artificial construct, it doesn't have to stay alive. And that allows me a lot more manipulation of that artificial cell. And then try to use all the tricks that a cell uses without all the complications of still being alive. And that technology could probably be developed. Now, it's going to be nanoscale technology, right? It's not going to be a big vat like this. It's going to be at the cell size level. And that's a few microns. And then the assemblies inside will be smaller. And that's going to require sort of nanotechnology. But what's wonderful is that's what's happening right now in the computer industry is nanoscale development. And so what often you see in technology is that you get to a place where you can't keep going, and some other field has just had a breakthrough or developed something that you say, oh my God, there's no way I could ever do all that, but they did it for another reason. And that's why you want to have good communication. And that's why I'm involved with lots of different departments, and David is as well, because you've got to bring in different technologies, try to solve problems. So no one ever talks about size, but I'm going to bring it up because of the fact it's actually a serious problem. Usually people don't talk about the really serious problems, as you may have noticed in the last month or so. So sort of the last problem, well, actually sort of two problems, but they're kind of the same problem. And that is just capacity. I'm going to make this DNA. I have commercial firms. They're going to make the synthetic DNA. How hard of a problem is that? Well, the capacity, that is, how much can I make? Most of these synthesizers that make DNA, most of them are 96. There are a few of them out now. We have one. Is 384. That is, it makes 384 of them at one time. And if it makes it 100 bases long, well, that's pretty good, right? That's 380,000 bases. Yeah, but I'm thinking 3 billion, right? That's off a fair amount. And I'd have to run that synthesizer day and night for a long time before I'd get there, right? So we're not quite there in capacity. Another trick is to use another technology that was developed for a totally different reason. And that is, instead of putting them in little individual wells, I can put them on, make them as little droplets on a glass slide. and I can put them a lot closer together, and I can make hundreds of thousands of them. Oh, that's fantastic. That's the capacity problem solved. But there's another problem. And that is, when I do this to solve the capacity problem, it's hard to make them longer because the chemistry is not as good, and I get more errors. So that often is what you see, too, when you're trying to develop technology. You solve one problem, and you resurrect an old one that you had solved already, and you get stuck. And so it's not quite clear yet how to get over the capacity problem. It's still being batted around. If I can do this way to solve the capacity problem and I can solve these problems, I'm there. Or maybe I can solve the number of well problems. The other problem that's associated with this capacity is cost. Now, as I said, if I had a spare $3 billion, I might put it into doing something heroic and synthesizing something big. But that would only give me a lot of credit. But it wouldn't solve anything, because the student that I'm training can't go off and do the same kind of thing. It's another $3 billion. Right? So to really make a breakthrough here, you've really got to make it easy. And that's what technology has to be focused on, not just making it possible to do something, but make it easy to do. So the whole effort here has to be to make it easy. And if it's easy, it will get used very effectively. And so cost is still a serious problem. So in a standard synthesis, those synthetic genes the raw base cost is about 8 cents a base. So that's not too bad. But if you want them to assemble it into a gene and make sure it's right, then it goes up to $1, right? Mm-hmm. So some people still go ahead and pay the $1 because they don't want to mess with it, right? But so that gets too expensive. In the arrays, it's quite a bit down. It's in fairly large synthesis on arrays, it's 0.1 cents, and you can in fact make even cheaper than that. So that gets pretty good. That's getting down there. Okay? So I said I would show an oligosynthesizer. So here's our latest oligosynthesizer. We hope to get it up and working in about 2 months. It's a breadboard instrument. See, that's why you can see all the wires, because you go in there and you do stuff. And then just shows you how, when you're developing a piece of a new instrument, this is what they look like. A mess. And fortunately, someone who actually knows how every part is in there works, so we can do stuff with it. And the synthesis is up here on this platform here. But this instrument is designed to make 3,450 oligonucleotides at a time. And it uses much smaller volume, and so our cost estimate on this one is around 10 cents to make 100 merc. So we've been working on a lot of the chemistry to get longer and higher quality synthesis. We don't know that this will compete effectively against the array methods. It's an alternative way, and we need to look at. It still has a capacity problem, because even this is not phenomenal. This is only, a factor of 10 over the others.
Now, would you say 3,450 at a time? How long is the time?
Well, you can do probably 3 runs. Well, for 100 mers, you can probably do 2 runs a day, right? So, and you might push some of the cycle times, but the problem is if you push the cycle times and reduce them, then your quality goes down. So it's always these, and if you don't need the high quality, then you will do that. And you can use less reagent to get the cost down. So all these are trade-offs that you have to try to play around with, which is your difficult step. It's conceivable that an instrument like this could be made that was even larger than that made more than that. But this keeps them in individual wells. So, and the reason we're not sure yet whether we have to do that because of all the assembly problems, or in fact we can get away with putting them all in one. This will make a lot more of individual one, and we don't need any other special methodology. So sort of that's where we are in trying to solve this problem in terms of a technical problem. We have a long, long way to go, obviously. But it's very valuable to know, what is it we're shooting for. So I think in the short term, what we're probably going to see is what's more feasible. So a mammalian genome has still a lot of problems left. What about a yeast genome? 12 million bases. Not so hard, not so expensive. A lot of other tools. So our plan is to make the yeast genome. Our plan is to work with someone like David and say, what does the ancestral sequence look like for yeast? And make that. So our first one we're going to try to make is we're going to go through and maximally change the sequence to try to find out how far we can push it away from the existing one. Now, once we've done that, and I think we can do that, then there'll be no relationship, because we've done it maximally, between those two. They won't recombine, they won't hybridize, and that means I have a platform that I can do other manipulations in, and I don't have to worry about the existing genomes being recombined and so forth. So it'll be the workhorse. It'll also be a new species, because by definition, it can't recombine with anything that exists. And that's the definition of a species. But it's really designed to be a workhorse to allow us to do lots of other things. And that's also what you see with technology, is that you bootstrap your way into it. You use one system, you develop it, and then you use that system to do the next thing. So that could be used to build the, say, bigger pieces for something like a mammalian project. But I think we have a number of real serious breakthroughs we have to do, or lucky. Sometimes you just get lucky, and you find that, oh, this is not nearly as hard a problem as I thought it was going to be. And that's how you bootstrap your way. So I'd like to stop there so we could have time for questions.
Well, thank you very much. That was nice and clear and simple. Thank you. Thanks, both of you. Could you both, I guess both of you will take a crack at each one of these questions here. I'm just going to ask, because I can, I'll ask the first question. Is it possible, combining chemistry and computers, to do that biochemical spell checker in this way? As you know, many people now are working on DNA computers, or using DNA to do primitive computer functions. Is it possible one day that we may use DNA to do error correction on DNA and make a biological computer do that?
I would say yes, but not with a DNA computer per se, but the kind of error correcting that the cell does.
Ron seemed to indicate that it's not a computer problem for the error correction.
No, I think that's Ron's problem. That's a problem he can solve.
I think it's just a biochemistry problem, and I think there are ways to do it. The cell has the same problem, and so you simply bootstrap your way into figuring out how the cell does it, and then use some of those mechanisms. So the problem is that a cell makes mistakes. A bacterial cell will make mistakes, and it has a way to go in and recognize those mistakes.
They somehow correct it.
And correct it. The biggest problem is that it doesn't know which way. It tries to use techniques that tell it which way to correct.
Okay.
And usually what happens is the new synthesized material is where the mistake will be made. And so what it does is it compares the new to the old. And then it corrects it to make it the old. And the problem is if it misses that, then it becomes the old, and now you're stuck. Right? So it's not perfect.
So nature makes mistakes too.
Yeah, nature makes mistakes.
I think I heard that Craig Venter's group recently made some kind of synthetic genome for maybe a small bacterium or something like that, and I was wondering if that was tested and it actually lived, A, and B, did that infrastructure or that lab or those people, are they likely to take off in this kind of direction and come up with something powered by industrial money that's going to overtake you, and you should just use them instead?
The future I don't know about, again. The problem, and I didn't mention it because I didn't learn anything from it. And I always I have to learn something. And the reason I didn't learn anything from it is that the genome is relatively small. It's in less than 200,000. So I know that I can physically deal with that. They never solve any of the size problems. And the other issue is how to do all these error corrections and so forth. Well, they simply bought all the pieces. They spent the $1 a base and ordered all the pieces, and all they did is stuck the pieces together and put it into a cell. Now, I already know that if I synthesize DNA, that's why I gave you those lessons. I know it will work when I go in. So there wasn't there was no hypothesis that I was concerned about for that experiment in terms of, oh my gosh, look, it works. Of course it works. That's exactly, now if it didn't work, I would have learned something. Now, of course, they couldn't publish it, but that would have told me a lot. Whoa, there's something we don't know. We better figure out what it is, right? So that's interesting. I mean, but you really have to learn something from it. And it was a spectacular experiment in a sense, but that's what it was designed to be. But it doesn't change the landscape because it's too expensive, right? So they didn't figure out a new way to make them cheaper. They didn't figure out a way to do error correction. They didn't do anything about the size problem. They basically took a problem that didn't need any breakthroughs, but only needed money.
These gentlemen aren't in a position to say this, but I will tell you that Craig Venter is a controversial figure. Okay, he's a very smart guy. He's a very talented guy. I don't want to diss him, but he's also a very good businessman and a very good promoter. I have a lot of respect for him, but you have to read his papers with a different eye. That's as much as I'll say here. Yes, ma'am.
Could you comment, please, on how chromatin and epigenetic influences generally influence what you're trying to do, if at all?
Well, you may have an answer to, and I have one.
Go ahead.
That's a fabulous question. In fact, that was one of my slides I was going to try to drop in, and I never had time to do. Because that's another problem we haven't solved. That is, we know that there is a lot of methylation of the DNA, and that methylation gets added afterwards. And there's a place on the DNA which you can put a methyl group chemically. It's just a chemical group. And that changes how that DNA works. And you can remove it again. It's reversible. And it's not encoded in the sequence. It's an add-on. And we don't know what the rules are, right? So we don't know if that methylation pattern is totally controlled by the DNA. So if I don't even worry about it, stick it in, it'll work fine. Or I actually have to come up with the right methylation pattern before I can make it work. Well, it's tough because I can chemically synthesize it with a methyl group on there. No problem. The problem is if I want to say, OK, I'm going to put it in yeast first, it's going to go away. So it could create a serious problem, and it could cause us a lot of the other technologies developed not be compatible with trying to get all the methylation patterns. So the methylation patterns are thought to be important for the development and things of that type. It's also possible that all I have to do is get that chromosome built. It's not going to be right methylation. Get it into a cell, get it to be carried for a few generations or even through one round of an organism's life cycle, and then it'll be OK. Right? So it's not quite clear. But the epigenetic changes that they're called could pose a serious problem.
Does David have
Absolutely, yes. You would have to go to a mammalian cell system, I think, and then, but the key idea is the last one that Ron said is bootstrapping it, right? You don't get it right the first time, but you've got to somehow get it through a generation or two and have it reestablish those epigenetic signals.
Okay, we have another question across the aisle here.
Oh, this is just a technology question because it seems like a nice machine you got there, but it's basically a printer. Why don't you just use a printer to, you can put reagents in the little things and a special piece of paper and print out billions?
Yeah, well, that is one of the mechanisms for what's called array synthesis, and that is it's done on not a piece of paper, but a piece of glass. The reason that they don't work as well is, one is you're synthesizing directly on the surface of glass. There's not a lot of surface area, so you don't get very much made. And it's very difficult when you have a picoliter drop to mix the picoliter drop and how to get the reactant, how to get it to react more efficiently. So it'll work pretty well. That's why you have more errors. You have a hard problem making sure that you get a very efficient coupling on that surface. But you're absolutely right. That is effectively the array synthesis, and then there's these complications. So the more conventional method, what it uses is a little glass bead that's very, very porous. So it has a very, very high surface area. And now I can mix the liquid around the bead and get a lot of mixing of the reagents near the surface. And that allows me to get more complete reaction. That's why it's a bit of a trade-off, right?
Okay. We'll have to move it along here. Question number 3.
Do you think it's practical to have an integrated circuit type of thing with channels in there with different chemicals to do the DNA synthesis and also to read the DNA to see if it was synthesized correctly?
Actually, there's microfluidics doing that here at Stanford, right?
Yeah, that certainly is a possibility. The reason that's not done on chemical synthesis really probably is because it's a lot of steps and they're complicated. And that just makes the architecture more complicated. And it's not totally clear that you gain a lot by having all these little channels. What you really want to do is minimize the amount of reagent that's consumed. And having a lot of channels that are filled with that reagent and you throw them away at the end consumes reagent, even at very small volume. So that's why this version is that you shoot the reagent to an argon atmosphere into the reaction chamber. And each reagent that you use has a dedicated nozzle, so that you don't, there's only thing that's wasted is the stuff that's right around the reaction volume. So what you're always trying to do is minimize the reagent consumption.
But
sequencing and things like that, that's actually is how people are now going to be doing sequencing. Sequencing is chemically a little simpler.
Apparently there are people in the audience working on the technology problem.
I'm sure that's probably true. There's, I know, 3 or 4 companies right around us that are doing this in different ways.
I have a question for David about the math. Both of you had mentioned earlier that there are many amateurs here who could do amateur DNA sleuthing, but they probably need tools such as software that you might be able to provide. Is there any public domain software that would assist in that kind of spelunking?
There are a number of different tools that you can get by just Googling one of the bioinformatics websites. There's a place called Pedro's Page, which you probably don't first think about Googling, that has all kinds of bioinformatics programs that you can download. But you can actually do a lot on the browser by just configuring it to show you different things. So you can almost do experiments live by saying, okay, now show me this DNA aligned with this DNA, and now
you mean right on your site
and so forth, right on the web. Yeah. So it depends on whether you want to write a program or whether you just want to kind of configure the DNA to explore it on the web without writing a program, and you can do both.
But it's not expensive. You don't have
they're
to
all
invest.
free.
Right. You don't have to invest in a lot of software.
No.
Right.
In fact, that's something people don't probably realize, that the Genome Project started this movement to make everything free and also make it instantly available. So when they were doing the sequencing, the rules for sequencing were that when I did a sequencing run, I had to release that sequence to the public within 24 hours. Now, that was faster than I could actually look at it myself.
Yeah.
And so everything was on the same playing field. And what you also don't know is an awful lot of other types of genomic data being collected, a lot of gene expression data, protein data. A lot of these that are done in very high throughput, often for a specific problem, but it's more efficient to do it all at the same time, and you're only using 1% of it in your experiment and publishing it. The rest of it's just there, and it's all you can obtain it.
Okay,
There's been a big push to make sure all the raw data is available, and that's now happening. Originally, people weren't releasing their raw data. Now they're releasing raw data. And there are companies that are going around and collecting it all so it's more easily available. Some of them, they'll charge you for it if you're a commercial entity, and it'll be free if you're academic or a student. And my guess is that you could probably generate a PhD thesis without doing a single experiment.
Seriously?
Oh, yes.
Oh, yeah.
That's absolutely true.
Hundreds, thousands of PhD theses.
We have time for 2 more questions. This is the penultimate one here.
Okay. I had a question. What is the time frame for these problems to get solved? I mean, what kind of time horizon are you looking for? 5 years, 6 months, 25 years? Or is it much even further than that?
Depends on money.
Oh.
So if we were starting the human genome today and the amount of government interest was the same as is on synthetic biology, we would be talking 25, 50 years. to get the human genome sequence. And it was only because it was elevated to a high priority, this has to be done. Money is available if you have a good idea. But the way a lot of stuff is today is if you have a phenomenally fantastic idea that's way out there, way ahead of its time, there is not a chance that you'll ever get funded. Right?
Philanthropy.
Great.
That's just the way it works.
Philanthropy could change that.
Yeah. So it's the funding that puts brakes on some of these things.
Okay. Okay, a couple more questions.
Just surfing on the web, I see that in 1997, a small artificial human chromosome was successfully synthesized, and it survived a number of cell divisions reproducing. And I haven't seen anything, I haven't been able to find anything about it since. And I wondered what happened to that. Do you know anything about that?
I don't know exactly what you're referring to.
Probably Hunt Willard's work.
Yeah, there are things which people were able to sort of isolate in vivo created molecules that they could create and passage them. They didn't actually synthesize them. They took something that actually existed. Or there are some other work where people have synthesized it, but now it's extremely small. And they're worried about having sequences that will replicate, sequences that will stabilize it. And they might call them an artificial chromosome, but they're very small. And they contain, 10 genes, something of that type.
But you can get a mammalian cell to carry an extra chromosome, and they do this with mouse, and they've done, Hunt Willard has done some with human cell lines. So it carries an extra chromosome. But it was, as Ron says, it's not fully synthetic. But DNA is the DNA, right?
Yeah.
So it will work as a synthetic chromosome as well.
All right,
That's why you think it's...
question, and then we have some announcements. Sir.
Okay, this was couched as a possibility to bring back extinct species, and I'm curious, what are some of the potential dangers of this technology?
Right, the T-Rex got loose in Jurassic Park. I'm not so afraid of crazy ancestral mammals running amok. Here, I think it would be very, very exciting if the technology that Ron described someday came to fruition and we could reconstruct a shrew-like ancestor and study it in the laboratory. That would be incredible to me. People would ask, do you want to reconstruct the human Neanderthal ancestor? And I think that would be quite a moral dilemma at this point, given that we don't know really whether we're getting these things accurate or not. The other end of the spectrum is that you could also reconstruct, and people have built dangerous viruses. And I think that is a technology that has to be carefully monitored. We don't want DNA construction for destructive means, and it's something that a society has to be very careful about, and I think we need careful regulation in that regard.
Yeah, the viruses would be the easiest to do and also the more destructive. So, and that could happen just accidentally. So anyone doing any kind of human or mammalian virus construction has to be quite careful about what they're doing.
Well, I think this has been, and I meant it earlier when I said you had both managed to simplify it a good deal, and I think you actually managed to answer the question and actually inspire some people here to go out on the web and do some serious research just with their own tools and your tools. Thank you, gentlemen, both very much. Ron Davis.